Okay, so welcome to Ask the Expert. We're very pleased to have Holger Russ with us today, talking about generation of stem cell-derived functional thymic cells for research and cell therapy, and just a little backstory about him, or a little bit of his bio, is that the emphasis of Dr. Russ' research is on understanding the underlying molecular and cellular mechanisms resulting in autoimmune type 1 diabetes, or T1D, in humans, with a focus on insulin-producing beta cells. During his career, Dr. Russ successfully worked on different aspects of T1D, which led to several original and important contributions in the fields of beta, thymus, and stem cell biology. After completing his diploma thesis in Heidelberg, Germany, he obtained his PhD from Sackler School of Medicine at Tel Aviv University, Israel, followed by postdoctoral work at the Diabetes Center at UCSF. He established his independent research program at the Barbara Davis Center for Diabetes at UC Anschutz Medical Campus, and recently moved to join the Diabetes Institute at the University of Florida, Gainesville, to further expand his research program. His lab employs state-of-the-art human stem cell technology and primary human cell culture with genome engineering approaches to model and potentially treat patients suffering from diabetes. Dr. Russ' long-term goal is to understand why T1D develops and develop novel intervention and treatment modalities, maintain a productive and extramural funder research program, and train and enable the next generation of scientists focused on T1D research. Welcome, Holger. Thank you very much for joining us. Sorry, I just had to switch screens. And we're really looking forward to this talk.
Thank you. Thank you so much for this kind introduction. Yeah, that's right. That's the goal basically of my research. So today we're talking about generation of stem cell derived functional thymic cells for research and also potentially cell therapy. And I see we're a small group. So if there's questions during the talk, feel free to just chime in. That might influence my view on things. So to start off, the thymus is probably this one organ. All I see here probably knew where it was and what it did, but a lot of people don't know that they had it. So it sits in the chest cavity right on top of your heart, and it's basically the key organ to establish your adaptive immunity and establish two things. First, positive selection, that T cells that come from the bone marrow migrate into the thymus, can recognize peptides mounted on HLA molecules and being recognized by a TCR. And that's really critical in sensing later on foreign protein peptide compartments and activate the immune system to basically recognize there's an infection. The second part is negative selection. Now, when a T cell can interact and sense, there needs to be a strain on if a T cell reacts too strongly with peptides that are presented everywhere in our body. And one of the manifestation of when this process can go wrong is autoimmunity, that basically a T cell responds too strongly to self, and autoimmunity ensues, like in type 1. So the thymus is really key in establishing a diverse, active, but self-tolerant immune system. As said, recognizes pathogens, but is tolerant to self. And there are forms which prove that congenital athymic conditions exist, resulting in T cell lymphopenia, where you see basically the absence of T cells, and therefore individuals are prone to infections and so forth. So I have a kit here represented. I hope you can see my cursor. So fully active in the young, but as we age and with clinical interventions, the thymus involutes, naive T cell output declines. As said, when you, let's say, get chemotherapy, this further accelerates this decline in naive T cell output. And we get over time, as an individual ages, a shift in the T cell phenotype from a naive to a more memory cell type. And with this, with impairment of the thymus or failure of the thymus, we have an increase in the risk for infections, increased cancer development, reduced responses to vaccines, and predisposition to autoimmunity. So, yeah, I don't have an organ anymore. Most of you probably either. Okay, so the question that we started off a long time ago, actually during my postdoc time at UCSF, can we generate a functional human thymus? And why would we want to do this? And just some examples, and that hopefully gives the idea why we have been so keenly interested in understanding how we can replicate probably development of the thymus in the dish, is that there are examples where we can use it. One can think of treating rare congenital diseases with just providing an autologous thymus, right, that provides at least partial reconstitution of the T cell education and adaptive immunity, and therefore provides protection and treatment modality for those babies. One can also think in cancer settings where there's a bone marrow transplant, where a lot of patients suffer actually from the lack of proper reconstitution of the immune system and therefore secondary infections that are very often lethal. One could envision that co-transplantation of a thymus to restore at least partial immune system, again, can have tremendous upsides and patient benefit. Then, of course, you can think of a more broad approach that we will establish a human model system to actually study thymic function and T cell development, and then probably can exploit this ability to generate specific T cell populations. We can also think of using this approach in a more broad regenerative medicine approach, probably in the allo setting, where we think of, okay, can we generate, in the sense of type 1 patients, can we generate a pancreas from a certain cell line and a thymus, and then basically transplant that into an allogenic recipient, but trigger basically re-education of the immune system present in this patient to allow tolerance towards an allogenic graft. And then in the long run, right, there's also this idea that if you can rejuvenate the adaptive immune system, can you rejuvenate an older person in general? So all, I think, very interesting and attractive approaches to why we want to do this. And how we started out is we used human embryonic stem cells, pluripotent stem cells. By pluripotent stem cells has been used in a lot of different settings, but basically two main attributes. One is that pluripotent stem cells divide indefinitely and rapidly, so we can make a lot of them. And if we know how, given the right cues, we can direct differentiation into any cell type found in the human body. And as you can see here, a large part of my lab focuses on pancreatic beta cell, but also thymic epithelial and some motor neuron work we do too. So initially embryonic stem cells, then with Yamanaka and Takahashi's discovery, right, we can do induced pluripotent stem cells through reprogramming of donor cells, and we can combine that now with genome engineering, which I won't go much into, but really allows us whatever you can think of engineering wise, meaning knocking out a gene, overexpressing a gene, putting SNP configurations in, making reporters, is all feasible now, which gives us really this toolbox. So if we can make a functional cell type reproducible, we can really drive the discovery process. So how did we go about when we started to formulate protocols? We basically looked in development. The thymus develops from the endodermal germ layer through branching, and this is just from Zorn and Jim Wells, where you can see that in the endoderm, in the anterior part, in the third pharyngeal pouch, the thymus originates. And so can we mimic this in a stepwise manner? And it's been done for many different tissues. This has been done in a stepwise manner, mimicking what we know in development, mostly from animal models, also a little bit from human development. And really the question is always, okay, we define, for example, here, pluripotent stem cell and definitive endoderm. We use markers that we know, that we trust, that we make the right cell type. And as you can see, there's just 3 stages here, actually, because we don't have particularly good reagents and a handle on really looking at what defines a thymus along here. Maybe some anteriorization we can still check. So what you need is HOXA3 is third differential pouch marker, I absent 1, and then thymic epithelial progenitor cells are defined by FOXN1, HOXA3, and I absent as well. So when we go, this is 14 days. So this work is basically based on the postdoc work, which was really led by Audrey Parent, and I see you're here, Audrey. Hi. In a stem cell paper using embryonic stem cells. But when I started my lab, we were really like, okay, we want to test if this protocol works on many different iPS cell lines that we generated, because classically, and this is not just for the thymus field, in many fields, is when a protocol is generated, it is kind of specific to a certain cell line, can usually be transferred somewhat, but there can be stark differences within pluripotent stem cell lines. So we set out to, can we use this protocol that has been published as a baseline, but then test a lot of signaling pathway modifications along the way, and do that in conjunction, in parallel with multiple iPS cell lines to really have reproducible protocol. And short answer is yes, we could induce, define a protocol that gives us thymic epithelial progenitors reproducibly. The expression levels are not as high as we wish, but reproducibly. During the course of this study, we also identified, as others have shown at that time, that CD205, a surface protein, together with AbCam, can be used to approximate thymic cells. And when we do this quantification, we can find that approximately 40–45% of the cells at day 14 adopt this thymic epithelial-like progenitor state, at least by the markers we use. So we established an improved thymic progenitor protocol that works on many different lines. We've done that now in dozens of lines. But can those thymic epithelial progenitors give rise to functional thymic cells? So to do that, we use the model system, which is the nude mouse. So the nude mouse has the T cell compartment, but doesn't develop an endogenous thymus. Now, if you take a fetal human thymus and transplant it into this mouse, the mouse T cells will migrate into the human thymus and be educated. So it's a great model to test the competency of those TAP cells. And we asked, do TACs, thymic epithelial cells, develop, and is there mouse T cell development? So staining for our thymic markers, we find within the grafts, within these human cells, AbCam CD205 positive cells, which indicate thymic. And we find developing TACs. Why can we say that? So TAPs in our system express keratin 8, which marks these progenitors, but also later, after further differentiation into TACs, marks cortical thymic epithelial cells. Keratin 5 is in this context more specific. It labels medullary TACs, and these progenitors going to become TACs go through a double positive stage. So when you look here, we find a lot of keratin 8, which is expected, but we have distinct areas of keratin 5 positivity and some cells that are double positive, indicating this is an actively ongoing further differentiation of those cells. When we take the grafts out and we do qPCR, this is also published. We did also a bulk sequencing. You can see that our key marker FOXN1 goes up. AIR, important marker for negative regulation, we will touch on later, does not change, but we got upregulation of keratin 5, keratin 8 stays the same as somewhat expected. And then other molecules important in T cell selection and attraction, chemokines, are upregulated in the grafts compared to our controls. So great. This data basically indicated we can make TAPs universally in vitro. We transplant them and they further differentiate into TACs. What about the T cells? When we look at the T cells, we find that there is areas of thymic cells surrounded by mouse CD3 cells. And we also find areas of developing T cells, meaning there are CD4 T cells and CD8 some of them are double positive. We can take the grafts, make single cells, and stain for CD8 and CD4. This is pre-gated on 3 and 45. And we find double positive cells. We can quantify that in many different animals. Also, what we did too is to define if these T cells that are formed are actually diverse in repertoire. We did this TCR sequencing of CD3 cells isolated from blood. This is graft-bearing, tap graft-bearing newt mice. You can see each bubble is basically a TCR. The smaller the bubbles and the more of them, the more diverse. This is just copied from their homepage. You can see in a wild-type mouse, it's very diverse. The diversity is enhanced compared to controlled newt mice, where we just have very fully understood but reported by others. There's a couple of T clones in the newt mouse which just expand. We don't understand really well where they form and how. Okay, so this system we can also educate in this stem cell derived thymus mouse T cells. Okay, now taking a closer look when we did these studies, what we did next is we did single cell sequencing. And what we did is we took grafts from 2 different iPS cell lines and we compared them to a primary neonatal thymus. That's the same thymus, it was just prepared in 2 different ways. What you can see is that we can identify cells nicely, they cluster. We can use just marker expression to define cell categories. Here are lots of T cells. Here is mouse host cells. And here are populations which we define as TAPs and TACs. And this is really the take home here is when we now look with the different colors, you hopefully can appreciate that the stem cell derived TACs mingle transcriptionally and are not distinguishable from the primary human TACs that we find in a neonatal thymus, both in this large population and this large population. There's also this population of only iPS-derived TACs, which are probably earlier in development, at least that's what we believe. But again, we generate with this system cells that are transcriptionally indistinguishable from primary cells. Okay, we were of course happy with this. This is a quick first summary. Development of an optimized direct differentiation approach that works on multiple human pluripotent stem cell lines. We think this is important. The process takes 14 days. We reproduced some aspects of this in 3D suspension-based bioreactors, thinking of upscaling. Those TAPs can further differentiate into TACs. However, this takes months, right? And it's still a xeno model. So at this point, we were like, okay, can we define conditions that generate a functional TAC in vitro, right? Obviously, next step. So we looked into the literature and people have shown that when they use fetal thymi and they grow them either epithelial enriched or mesenchymal enriched, expand them and then reaggregate them in an air liquid interface together with cord blood derived hematopoietic stem cells, they get some function of the thymus back, indicating that these culture conditions would support the function of a mature or functional thymic epithelial cells. So we took exactly those culture conditions and just took our TAPs and put them in this air liquid interface. And we basically assessed, do we get further maturation of those cells? And we used again the keratin 5 as a marker of medullary, CTACs, and development. And I hope you can appreciate in the TAPs here, we only have the progenitor markers or CTAC marker, but in the STOC after 2 to 3 weeks, we get pockets of MTAC-like cells and double positive cells, which indicate active development. Now, I want to point out, this is in the absence of T cell progenitors, which is by itself interesting. So this is pretty easy system. So we also did some flow-based quantification. This is TAPs, this is STOs at 2 weeks. There is class 2 expression, which is low, goes up. It's all against AbChem. There's the thymic marker, which is lower, goes up. And then we use a stem cell line that has a GFP knocked in one allele of the FOXN1 endogenous gene, and we can read out GFP which in these experiments also goes dramatically up. So all in line with showing further differentiation into the phenotype we want to have. However, when you go and you define based on markers, right, it's just a like cell, and the ultimate test is function. So we were thinking, okay, how can we introduce the components to test function, meaning T cell development in the system? So we turned to literature, and Ed Stanley group recently published a protocol that generates hematopoietic lymphoid stem cell progenitors that are marked by CD34 and 45. And then Jim Welf's group published a protocol a while back that allows the generation of splengenic mesenchyme. And so through literature, okay, we needed the T cell compartment, but also there's a large body of literature that shows that mesenchyme has an important role in supporting the epithelial compartment of the thymus. So we set out to reproduce some of these studies I just mentioned, and we can effectively make CD34 and 45 cells, we can freeze them, and we can very effectively, 96%, make mesenchymal cells, at least by PDGFR. We have some more data on this in the paper. So then when we go ahead and we co-culture them, and this is now compared to primary human thymus, we get areas of keratin 8 and keratin 5. Overall, these are not as structures that we would like to see. It's still thymus-like. It doesn't look like a thymus, unfortunately. But the marker expression that we would like to see is there. We can, of course, look at qPCR, and we find that actually FOXN1 is in this setting not upregulated. But I want to point out that this is basically now 3 types comparing a multicellular organoid to the original TAP. So there is probably enrichment. Probably an underestimation. So keratin 5 remains same, but keratin 5 goes up, as well as DLL4, important for the specification of T cell progenitors, and class 2 backing up our flow-based data shows that there is further maturation within these stem cell derived thymic organoid cultures. Now, what about the T cell compartment, right, which we really care about? So we can gate on CD45 and then on pre-pro T cell progenitor markers CD5 and 7. And from there go into, do those develop into 8 and 4. And as you hopefully can appreciate, as again, primary thymus STOs, we get a double positive population as well as CD8 and some CD4. CD4 is in general not as effective as we see in primary human thymus. We can, of course, also gate on TCR rearrangement and surface as well as CD3. And when we do so, it's even more evident that there are differences, but we get generation of the cell types that we would like to see, which indicates that, yes, we generate functional tags in vitro. Now, with the emergence of CD4, we were also very interested in further defining it. And when we stain some of these STOs, we find air protein. This is in certain areas, it's not throughout. And we also have some samples that are negative. But in the ones where we find it, we've done a lot of these stainings. It is present. That goes together with some of the qPCR data we have, where air is critical for tissue-restricted antigen expression, sporadic in thymi. And we find from different germ layers, TRAs expressed. So indicating that these STOs have some features key in negative selecting of potentially autoreactive T cells that might develop in the organoid. Okay. Other work has shown that negatively selected, or T cells that are developing and undergo negative selection, might express PD-1. And we find, although at very low levels, we find CD3 positive cells that also express PD-1. This is CyTOF data, where we collaborated with Dirk Hohmann at Mount Sinai, where we find some PD-1 positive cells if we gate on CD45, 33. Similarly, when we look at CD4, CD25 gated, either in primary thymus, STOs, isotype, or FOXN1 positive cells, we find FOXN1 positive T cells within, which further indicates that there are features of negative regulation. To further support this, we also did qPCR, and while in the thymus, this is 2 orders of magnitude or something higher, FOXP1 expression, we find specifically FOXP1 in STOs, while we don't see this in TAPs, as is expected. So with this, I hope I could convince you that we developed a STO system that supports TAC and T cell development, and that it exhibits key markers of negative selection. So that kind of now gives us a handle to address a lot of questions we have and we're interested in. So can we combine genome engineering, right, to study biology in a strictly human isogenic context? Can we improve on existing humanized mouse models when you think of, okay, can we now really start to provide in an autologous manner at least parts of an immune system in transplantation settings, cancer settings one could think of? Can we develop really like multiple organs on a chip, thinking of microfluidics, right? Then, of course, there's a lot of thinking. Can we improve actually STOs by improving the cell we put in? I showed you earlier on that we, with the Fox and one GFP reporter, we only make like 12% of the cells we actually want to make before we go in STO. So there's a large room for improvement there. Then, of course, can we explore orphan disease in an autologous setting? And can we think of strategies allowing targeting of a wider patient population, that you can move away from the individualized, more to an off-the-shelf approach that could help more people at a more affordable price? Okay, that's my new lab at UF, where I recently moved. This is still at the old place, and really the key players, Stephen Ramos, who's now doing postdoc with Sung Kim in Stanford, and Lucas, a postdoc who is actually went into the industry now, and undergrad student who helped throughout is now in NYU. They have been really pushing this, those 3 people, the whole of the project. Collaborators, of course, I want to thank organ donors and their families, funding, and you for attention. And if you're looking for a fun postdoc or any other position, contact me. I'm happy to take questions. Thanks.
Thanks so much. That was an excellent walkthrough of some really detailed protocols you guys have been busy building. I'm kind of curious whether, oh, here's a bunch of questions. I know we have the CSO, Dr. Bing Lim, in the audience, and I wonder, what if he has anything to offer, because their mission statement seems to be well aligned with some of these protocols you're building, as well as, they're targeting off-the-shelf solutions. So I didn't know if he wanted to unmute himself and ask a question.
All good. Can you hear me?
Yes.
Yes, loud and clear. How are you?
How are you, Holger? Beautiful work.
Thank you.
I mean, the in vitro work is really, really interesting. And the protocol you have is also very rapid. And I'm very curious. You're able to get the cells in 14 days?
Yeah. We are not, or my students and postdocs have not been super strict at sticking exactly to 14 days, just out of convenience, because so much is going on. But we can do it as early as 14 days. You can go up to day 19. We usually don't try to go longer, but yes.
And the CD205 is a very consistent marker for you to get the cells in the end?
Yes, we have been using it quite a bit. We actually haven't done anything of, sorting out and seeing which different populations to put together. Do we get increased efficacy or not? Yeah, so yes. I mean, you maybe have the bandwidth to do something like that.
Yeah, I really I think it's a very important advance you have made in the in vitro. I think your idea of testing the function in vitro rather than putting into the complex in vivo system, which takes several weeks to emerge, as you know, from the previous work. And this is really an excellent way. And I'm really looking forward to see as you add the different cell types and the modification, whether the efficiency will increase. And of course, you know, there's a lot of interest at the moment in this in vitro generation of T cell. Interestingly, you also observe that CD4 is more difficult to obtain. And
yeah.
do you have any
It's
thoughts on in the system? Any thoughts on why the CD4 persisted to be more difficult to obtain?
Yeah, sure. I mean, it's all class 2 based that we just don't get enough class 2
Do
expression.
your cells express? That's what I was asking. Do you know whether
So
your cells express class 2?
You know, it is in I think a lot of the approaches have been plagued by that oh sorry,
Limitations, yeah.
Flow with Austin, have to train my graduate student flow and stuff. Anyhow, what I'm saying is that, you know, we still see our differentiations are still not as consistent as we would like to see. And of course, I also think that this reminds me a lot of maybe 10, 15 years ago in the pancreas field, where we had some really encouraging data, but it was just a lot of experimental paradigms to test were still very challenging because we didn't have the consistency and the high level production of this on target cells, right? And I think in the thymus we're hitting similar challenges right now. So what I'm saying is that I do think that a lot of optimization can still be done, and that will really accelerate the downstream approaches. But I do think that two key challenges of the systems are the class 2 consistent and high enough levels of class 2, but then also committing HSCs towards a pre-pro T cell fate. I think those are the two key problems we face, right?
Yeah, I agree with you. Very nice work, and I look forward to see your further improvement on it. And I'll get in touch with you. We can talk a bit more. Thank you so much.
Sure. No, I would love
wonderful,
to. Thanks,
wonderful
thanks,
presentation. I'll yield the floor to other questioners. Thank you.
Fantastic. Love to hear that crosstalk. We also have a question from Albert Waugh. He's at the NIDDK. He's a program director in the Division of Diabetes, Endocrinology, and Metabolic Diseases there. His question is, how many cells are needed to get the systems to work? And do only a subpopulation of TECs carry out the selection? Do the T cells escape negative selection simply because of the ratio of cells? It's a nice technical question. Thank you.
Yeah, awesome. All of them are relevant. So how many cells are needed? We frequently do it in 24 wells, so it's low millions and you can get things going. There's actually, I haven't thought of it extensively, but right, how do you do air liquid interface in a larger scale? And we haven't experimented with this a lot. But so what I'm saying is cell number needed is relatively low. It's a bit of a pain to align all the timing. So we were also investing a little bit into freeze thaw to make it easier on ourselves. So then only subpopulation of tags carry out the selection. Likely, yes, it's still, not as pure. It doesn't look like a full-fledged thymus. So for sure, it's subpopulation driven. How this exactly works within thymic compartments, we don't understand too well. A hint onto Park et al., right? The science paper showed beautifully using single cell transcriptomics, and actually also Audrey subsequently, that there are subpopulations of tag cells. And when we compared the published data from Park to our in vitro generated stem cell tags, we find at least most of those subpopulations as well. So in a sense, it's like, I would say, probably nature does its thing and generate what is needed to fulfill function. We certainly have ways to go to further improve that. Do the T cells escape negative selection? Yes, very interesting, right? We have a lot of ideas how to approach those questions. And of course, it's like mind-boggling when you think of the millions of cells going through a thymus and being selected against, right? So how is just the interaction of stoichiometry working? I think it's not very well understood. We have some ideas how we can use transgenic, or not transgenic, but TCRs that have been defined by our colleagues, right? I think in the autoimmune space and also cancer space, how we can approach some of these questions just to understand, hey, when do you overload and negative selection just doesn't occur anymore because the interactions are not given on a numbers game. Yeah, it's all relevant and good questions which are on our mind. Audrey.
Yeah, we just have Audrey Parent here from UCSF.
Hey, nice talk. It's really great to see the progress in these newer version of the differentiation protocol. So my question is about your air expressing cells. I'm actually curious to know if you saw them when you didn't have the hematopoietic stem cells in there. We've been thinking for a long time that we need this crosstalk between T cells and these TEPs to get them to express air. Is that something you saw in your system?
Yeah, we haven't looked really hard. We were actually very surprised that we see this differentiation without the need for any T cell interaction, right? Like we just put them on this air liquid interface and they continue down their path. Now, again, we were surprised, right? And we see 5 and some other markers when we look at PCR. That was good enough for us. We were more like, okay, let's focus on the function, right? Because this is what people will ask us. So the answer is, I don't know conclusively because we haven't looked very hard. The air, that's why I pointed out, while we can find it in 60% or so of all STOs, you need to look.
Okay, but you didn't look necessarily in the TEPs only without the...
Nah, we didn't we didn't make a massive effort to look, no.
Okay.
Yeah, a super interesting question, of course.
So many things to do. Just sort of like last question, I wondered if, just want to do a little thought experiment and sort of, kind of cast that thought experiment towards the clinic. How would you, imagine this kind of, off-the-shelf version of these cells being engaged in the type 1 diabetes realm?
So in the type 1 space, it's complicated, but overall, you can think of certain modifications to just reduce the complexity of mismatching or matching, right? Does that make sense?
Yeah. Yeah. And then, together, I don't know, maybe together with, in collaboration with others, you can get to that point of some kind of off-the-shelf solution. So
yeah, at least I don't, when I say off the shelf, it doesn't mean necessarily that it needs to be off the shelf for everybody, but if we can hit, like, let's say, a certain percentage of the population, right, that would be already great. Yeah,
I mean, if you could hit certain endotypes, right, during the prodrome, that would be phenomenal. So here's hoping. Just, lastly, thank you for this wonderful talk. In the interest of time, you had a lot of support in the chat. People thought it was wonderful. And just one more shout out. It looks like you're looking for, I think you said in the beginning, some postdocs. So if those are people are interested, this is some phenomenal work and really exciting. So I guess reach out directly, correct?
Absolutely.
All right, then. Okay, thanks again. Have a great rest of your day.
Thanks so much for having me. That was fun.
Great.
Thank you, Holger.
Really great. Thank you.